Because you need everyone. Oh yeah. I don't know but I thought the same way. I was like,
Aaron, they're dinosaurs. My good luck.
you wore a dinosaur shirt on Monday.
Did I?
Yes, you did.
What did I wear?
You wore a dinosaur shirt. Just wore a dinosaur shirt. You're amazing. So chic. I'm not anywhere anything.
Why are there so many people here?
Because this is important information that people want to know
All right, so what I'll do is I will introduce you. Get out of here. So it's about 10:30, so maybe we should get started. I'd like to thank everybody for coming to Alana's thesis defense. As I think most of you know, my name is Todd Waldman. I am Alana's thesis mentor, and I'm just going to sort of fairly briefly introduce her to you and then let her tell you about what she's done. So just very briefly, Alana grew up in New York, and she graduated from NYU in 2009, majoring in chemistry. After she graduated from NYU, she worked in several laboratories, first at a pharmaceutical company called OSI, and then at Cold Spring Harbor Laboratory. She then applied to the MD-PhD program and was accepted and matriculated in 2012. And I should mention that in about 2000, I took over as director of the MD-PhD program, and Alana was, I think, in the very first recruiting class that I did. So she was one of the very first students that I participated in recruiting. So she did the first 2 years of medical school and started in my lab for her thesis in 2014. So Alana and I have been working together for the past 4 or 5 years. And over the course of her thesis in my lab, she's won a number of grants and other awards, and I'll just mention some of them. She has gotten funding from the TL1 Translational Biomedical Training Program for 2 years. She was recently awarded a fellowship from the Mistletoe Foundation. She was awarded the Georgetown University Graduate Student Teaching Assistant Award, something known as the 3-Minute Thesis People's Choice Award winner, which I didn't know about until yesterday. She has been awarded a number of travel and course grants from the Medical Center Graduate Student Organization and was awarded a Blue Ribbon poster at the ACTS National Conference. And just a little bit of stuff about Alana's project. So when she started the project, which eventually became her thesis project, I think that we were all a little bit nervous about it because it was so different from pretty much everything else we do in our lab. So our lab is a pretty hardcore molecular biology lab. We do a lot of cloning. We do Western blots virtually every day, and a lot of tissue culture. But we almost never do immunohistochemistry, and we'd never before done anything like a biomarker validation project. So that meant that in order to successfully perform this project, Alana had to develop a lot of stuff that we'd never done before. And so she would have to establish relationships with all different kinds of people at tumor banks, both in the DC area and literally internationally, including tumor banks at Georgetown, at MedStar Washington Hospital Center, in Hackensack, New Jersey, and actually in Denmark. And she would also need to establish a relationship with our immunohistochemistry core here, which is run by Deb Berry, and I should just mention, does an amazing job. And also, we don't really use biostatistics in our lab very much, although I know we're supposed to. So she had to develop relationships with a number of different biostatisticians to help us analyze the kinds of data that she was developing. So she did all these kinds of things and more, all new things for us. And the most important part of it is that, as you can imagine, there were many frustrations along the way because that's just kind of the nature of science. And she stuck with it and made it through to the end of this project and validated what we hope is an important new biomarker for bladder cancer. So I'll just end with this. And when I talk to new graduate students, which I do, fairly routinely because I run the MD-PhD program, what I usually say to them is that my hope for you is that you make a discovery. And I don't really care if it's an important discovery or a less important discovery, just something new that is actually true. Because that is brutally difficult, but incredibly exhilarating. And she's done that. She's discovered and developed a new biomarker for bladder cancer, which shows real promise for helping actual human patients. Science is literally the only job that I know about where the only way to succeed is to do something that nobody has ever done before. And using that really tough measure, I think Alana's done that. So I'm really proud of the work that she's done, and I'd like to present her for her PhD thesis defense.
All right, well, I'm very happy to get going, very happy to be here today to show you the culmination of my work, especially since a lot of you guys came back after data meeting. In the interest of time, I will ask that you hold questions for the end. I will do my best to keep this to an hour, but it's my time to shine, so no guarantees. All right. So to get us started, we'll discuss a little bit about bladder anatomy and bladder cancer and how we discovered STAG2. Then I'll talk to you about our clinical project wherein we validated STAG2 as a biomarker in non-muscle invasive bladder cancer. And then we'll go through some of the characterization work I have done to try and understand the role of STAG2 in bladder cancer. And finally, a few concluding remarks just to wrap it all up. Okay, so you may hear me use these terms interchangeably during this presentation. Urothelial carcinoma, transitional cell carcinoma, non-muscle invasive bladder cancer. In all cases, I will be referring to papillary non-muscle invasive bladder cancer. I may slur these words a little bit. I'm not drunk. I'm really nervous. All right, so let's get right to it. All right, so here's the sagittal or midline view of the male pelvis. So the bladder sits right in your pelvic region, surrounded, as you can see, by a host of very important structures, and is a holding reservoir for urine. So here's a gross depiction of the bladder itself. The ureters bring urine in from the kidneys, where it is stored in the bladder, and finally released through the urethra. It's also surrounded by the detrusor muscle, which is a smooth muscle that helps the bladder to contract and expand in order to expel urine. So if we head down a level and now look at the microanatomy, so what we see here, this would be facing the bladder wall, facing inside the bladder. And that's lined up by the transitional epithelium, and just followed by the lamina propria. Together, these make up the mucosa. Underneath that, we have the submucosa, then the detrusor muscle, and finally the adventitia, or fat. And this is what it looks like under a microscope. On the left panel is what we just looked at in that diagram, and on the right panel is a close-up view of that transitional cell epithelium. This is a special epithelium that's stratified, and cells can actually descend and stretch, which makes sense when you think of form for function, in terms of the fact that the bladder is a holding reservoir. And it is the cancer of these particular cells that we will be discussing today. All right, so a little bit about bladder cancer background and some basics to get us oriented. So bladder cancer is actually the 5th most common cancer in the US. Males are more affected than females, whites are more affected than blacks, and this generally targets an older population. It is the 4th most common cancer in men, and the 8th most common leading cause of cancer-related death in men. So this year alone, we expect about 80,000 patients to be diagnosed with bladder cancer, and over 17,000 patients will die. So the thing to put into perspective, what I always like to say is by the time I'm done with my spiel in about an hour, 9 patients will have received this diagnosis and 2 will have actually succumbed to the disease. So it really is quite a serious problem. And despite that, research in this field is actually grossly underfunded. Now at diagnosis, about 3/4 of bladder cancers are non-invasive, and 90% of them present as transitional cell carcinoma, which is the cancer of that epithelium we just looked at. And again, just to get us all on the same page in terms of some vocabulary, pathological grading is determining how ugly are the nuclei. So we divide that into low-grade tumors, which encompasses pun lumps, papillary urothelial neoplasms of low malignant potential, and low-grade tumors. And then we also have high-grade tumors, which encompass tumors that are purely high-grade and those that have any area of focal high-grade. And in this study, we are looking at both high-grade and low-grade tumors. Staging considers how deep is the invasion. So in non-muscle invasive bladder cancer, we have the stages PTA and PT1. In PTA tumors, there is no lamina propria invasion, whereas in PT1, we do see invasion to the lamina propria, but not into the muscle. Whereas with muscle invasive tumors, we have stages PT2 to PT4, and those are various levels of invasion into the muscle, the perivascular tissue, and then finally into the surrounding organs. And lastly, another important thing to consider is that we have something called the dual track development in bladder cancer. So tumors arrive via 2 distinct pathways. We have the flat cyst tumors that have frequent P53RB and TERT promoter mutations, and they're chromosomal mess. And then we also have papillary neoplasms that have frequent FGFR3, STAG2, PI3CKA mutations and are chromosomally stable. And in this study, we're looking at papillary tumors. So to put that into some context, these are what those tumor stages look like on top of the actual bladder lumen. So here again, we're considering the PTA tumors and the PT1 tumors that do not invade into the muscle. So if a patient is diagnosed with bladder cancer, and yes, this is my slide to make sure everybody's awake, they get what's called a turbit, a transurethral resection of the bladder tumor. So this is done under general anesthesia. A patient has a scope inserted into their bladder so the surgeon can visualize inside, and then the tumors are resected. Often patients will also go for a restaging turbit to make sure margins are clear and ensure that staging and grading was done properly. Okay, so I told you that most lesions present as early resectable stage lesions, and if we know we can remove them, and if we know they're early stage, why do we even care? Why am I up here? I'm up here because the majority of these tumors will come back, and a lot of them will come back angrier. The issue is we don't know which patients will fall into which categories. And so what that means is we do tend to take a more aggressive treatment approach to ensure that we don't end up in the bucket with a poorer outcome. So the goal of non-muscle invasive bladder cancer treatment is successful minimally invasive tumor resection with organ presentation. And so in this case, you may or may not receive chemotherapy and or BCG immunotherapy. And the risk the way your risk is determined with a number of various parameters. We use tumor multiplicity, staging, clinical grade, et cetera, factor into these risk scores, and that will kind of tell you which treatment paths you'll go through. You will also receive many surveillance cystoscopies. So these are similar to the turbid in that you are inserting a scope to visualize the bladder. You are under local anesthesia in this case, but the issue with this is that these cystoscopies are fairly frequent. Depending on what risk category you fall into, you might be looking at a few every year for several years, and then they'll taper off. But this is actually why bladder cancer is the most expensive cancer to treat, period, full stop. It's a huge economic burden, and then moreover than that, from a patient care perspective, it's, if you could avoid this, I think we'd all agree you'd want to. So if the patient progresses and we can't preserve the bladder, we obviously have a lot more problems now. So first of all, we're looking at chemotherapy for sure, and most likely radiation therapy. And then if we can't preserve the bladder, we're looking at something called radical cystectomy, whereby your bladder is actually removed. And as you can imagine, creating a whole new bladder from scratch in patients who are generally older comes with a host of issues. And this is actually one of the things I always like to tell people is these surgeries actually carry a higher mortality rate than any open heart surgery. So hopefully I've convinced you that this is something we need to try and fix. And this is really the clinical problem that we're faced with right now. So let's consider it a little bit more simplified. So we know a bunch of people will recur and or progress, but we don't know who. So we err on the side of caution and intensely surveil these patients and err on the aggressive side of treatment if there's a question, because better safe than sorry, right? But better safe than sorry can lead to a lot of unnecessary treatment. And if you were certainly in this position, you'd want to know, what are my possible outcomes. So in an ideal world, we'd be able to stratify these patients based on risk. And in theory, patients who are higher risk would perhaps receive more surveillance and more intense treatment regimen, whereas those that are lower risk, we can maybe have more of a more watchful approach and maybe quit poking them so much. So I set out to find that biomarker. So let's bring it all full circle now and bring it back to STAG 2, since we're all urologists, and bring this whole story together a little bit. All right, so cohesin is a ubiquitously expressed multi-protein complex whose canonical role is to aid in sister chromatid cohesion. Cohesin also aids in chromatin structure maintenance, DNA repair, and can also influence gene expression. So here you see the four core subunits, SMC1A, SMC3, sorry, my arrow's not working too well, RAD21, and then either STAG1 or STAG2. STAG2 we find hangs out along the centromeres, where STAG1 likes to hang out on the chromosomal arms, or rather, I'm sorry, the telomeres. All right, so in 2011, Todd's former student identified recurrent inactivation mutations in STAG2 in a variety of human tumors. He then went on to identify STAG2 as one of the most commonly mutated genes in non-muscle invasive bladder cancer. And in fact, it was more common in non-muscle invasive disease compared to muscle invasive disease, suggesting that tumors with mutant STAG2 genes were less likely to progress to muscle invasion. And here's a representative image of some of the IHC staining that David had done. So down here, these are two samples that stain positive for STAG2 and their associated H&E images. And here's a representative example of a sample that lacks STAG2 expression. So there's a few features of the STAG2 protein itself that actually lend itself really nicely to an immunohistochemistry assay, and it results in an assay that's actually very robust and very easily interpretable. So first of all, STAG2 is highly and ubiquitously expressed in somatic cells, so it's easy to see, and by definition, any loss of expression is aberrant. It's also on the X chromosome, so inactivation of STAG2 only requires a single mutational event. So it's haploid in males and functionally haploid in females. And the key with this is that it results in a binary assay. Finally, 90% of tumor-derived mutations are truncating mutations, and so which results in nonsense mediated decay. So if we probe with the C-terminal antibody, we can exploit this feature and determine which samples have STAG2 inactivation. And these are just some of the different tumors we see along STAG2. And then importantly, we also have a built-in positive control in this assay. So again, I said all normal cells should stain for STAG2. So if we have any infiltrating non-neoplastic cells in these samples, they should by definition stain positive. And if they don't, we render the sample useless, and we can move on and restain and ensure that the assay actually worked. David then went on to find that tumors that are STAG2 deficient recur less frequently. So he determined STAG2 expression status in a panel of 34 papillary non-muscle invasive urothelial carcinomas treated with turbit, and this was a panel from MD Anderson. He saw that STAG2 loss was significantly associated with increased disease-free survival. In other words, patients with STAG2 mutations have a better prognosis. He also saw that only 1 out of 8 of the STAG2 mutant tumors recurred, whereas over half of the STAG2 expressing tumors recurred, and 4 of them actually metastasized. So on the biomarker trail that I started this journey on, it seemed like all signs pointed to STAG2. We had some biological rationale, we had an assay that was very feasible to use, and we had the clinical rationale. So for these reasons, I hypothesized that STAG2 is a clinically useful biomarker of recurrence in non-muscle invasive bladder cancer. So initially, I set off to validate STAG2 as a biomarker in recurrence, since that's the preliminary data we had. In order to do this, I collected all of the bladder cancer tumors housed at Georgetown University. After going through cohort discovery and our various inclusion-exclusion criteria that you can see on the right, we wound up with a final cohort of 82 patients. Here is our table 1 describing the cohort characteristics of the Georgetown cohort. I want you to note that we do have a higher male representation and a higher white representation, which is what we expect to see in this demographic. And also, we did see that low-grade tumors did seem to correlated significantly with STAG2 deficiency, and this actually confirms some previous work by other groups. So all good on this one. So then we went along and stained all of these tumors, and we found, as expected, that many tumors displayed loss of STAG2 expression. So on the top panel, we see tumors that stained positive for STAG2. So 54 out of 82, or 66% of tumors did in fact stain positive. 19 out of 82, or 23% of the tumors stained negative. And again, you can see our positive controls on here with these infiltrating lymphocytes really nicely. And then finally, we have mosaic tumors, 7 out of 82, or 11% of them stained with areas of patchy, discrete areas of STAG2 negativity interspersed with STAG2 positivity. And another thing that I want to note with this particular assay is that we had incredible agreement with the pathologists who reviewed these slides. So it really is a very easily readable and interpretable assay. So then we went and performed a univariate analysis to evaluate whether STAG2 staining was significantly associated with the current recurrence. And initially we evaluated 3 groups: tumors that were positive for STAG2, tumors that had uniform loss of STAG2, and then those mosaic tumors. So in terms of recurrences, for the positive tumors, 52% of them recurred. For the negative tumors, 22% recurred, and for the mixed or mosaic tumors, 26% occurred. And this relationship was statistically significant with the chi-squared test. And now when we plot this on a Kaplan-Meier curve, we actually do see that we also have a statistically significant difference in the separation of the curves. So again, we see in the yellow on top is the STAG2 mosaics. Right below that, these are the STAG2 negatives in green. And these are the STAG2 positives in red. So again, you can see that tumors deficient in STAG2 have a more favorable prognosis rather. So then we considered collapsing all of the STAG2 deficient tumors, the negative and the mosaic tumors, into one group, and found that it strengthened the correlation. So there's biological justification for this. So we know that bladder cancer tumors have been shown to display intratumoral heterogeneity, so it wasn't expected that we would see tumors with mosaic staining. And we've actually previously shown that tumors with mosaic inactivation of STAG2 harbor inactivating mutations of the gene in the STAG2 non-expressing cell. So biologically, we felt that this was justified, and then when we actually performed a statistical analysis, it did actually show improved model fit as well. So in this case, again, 52% of the positives recurred, and of the STAG2 deficient tumors, only 25% of them occurred. And again, this was significant by chi-square test. And we had model improvement in this Kaplan-Meier curve. So again, you can see we have a beautiful separation of these curves in terms of probability of recurrence-free survival, whereby the STAG2 deficient tumors fare better. Yep.
You see more women in your mosaic population?
People have asked that because of the bar body. I don't remember exactly if there was a higher representation in that sense, but we did see a trend in this cohort, and actually a statistically significant trend in females and STAG2 inactivation. So there, yeah. So next we tested if STAG2 was an independent predictor of recurrence in a multivariable analysis, and we were very pleased to find that, in fact, it was. Patients who stained positive for STAG2 are about 2.5 times as likely to experience a recurrence as those that are deficient in STAG2. I do want to note that due to the limited number of patients in this cohort and issues of data sparsity for some parameters like smoking, tumor size, multiplicity, other things that we think are really important in terms of bladder cancer etiology, we weren't able to include them in this analysis, but we did rectify this in the cohort I will show you next. Okay, so according to my reviewers, an ideal biomarker would also predict progression. So we worked really hard to try and figure out how can we answer this question. The issue with the Georgetown cohort was that only 6% of the patients progressed to muscle invasion, so there's just no statistical power there with which to conduct this analysis. But the science gods aligned, and we were put in touch with this researcher, Lars Scott, over in Denmark. And he had some fantastic tumor microarrays that were incredibly well clinically annotated, and was super, super excited to help us, and generously sent us these TMAs over. So the original TMA comprised of 289 tumors. I'll show you our cohort in the next slide. We've ended up working with 253 tumors. Of those 253 tumors, 93% recurred and 41% progressed to muscle invasive. So now we have a cohort that we can work with. So again, here's our cohort discovery flow chart. At the end, we ended up with, like I said, 253 patients. And in this cohort, 74%, or 186 of the tumors, stayed positive for STAG2. 24%, or 61 of the tumors, stayed negative, and 6 tumors, or 2% of all tumors, were heterogeneous or mosaic. And then here's just a scan of the TMA and some beautiful images that Deb had scanned for us. Here's a positive example, another mosaic, and then a negative example. They don't show as nicely on here, but they're gorgeous, gorgeous images on my computer, I promise. All right, cohort characteristics again. So in this cohort, I just want to note that we just went right to collapsing the groups to negative and mosaic. STAG2 status. And interestingly, what we found here, kind of alluding to what Darius had said, is that there was a statistically significant association between female gender and lack of STAG2 expression, and also with stage and lack of STAG2 expression. In the Georgetown cohort, we saw these trends, but they did not reach significance. And in the Aarhus cohort, grade was just barely missed statistical significance in terms of correlation with STAG2 status, whereas in the Georgetown cohort, it actually was significant. So we do suspect there might be some more to this story here. All right, so similarly to the Georgetown cohort, I then went and performed a univariate analysis to evaluate whether STAG2 staining was associated with progression in non-muscle invasive disease. So at 5 years, 38% of the patients who stained positive progressed, whereas those that were deficient in STAG2, only about 16% of them progressed. And again, this correlation was statistically significant. At the duration of, at the culmination of the follow-up at about 12.5 years, it was 45% of patients who were positive for STAG2 had progressed, whereas only about 16% of patients who lacked STAG2 progressed further. And again, we were very pleased to find that STAG2 is an independent predictor of progression in this cohort. So in this case, we found that patients who stained positive for STAG2 were nearly twice as likely to experience a progression. All right, so just to wrap up this first part, so we've shown you that a simple, robust, reliable, and importantly, binary assay can identify STAG2 mutant tumors. And patients with STAG2 expression are about 2.5 times as likely to experience a recurrence, and about twice as likely to progress as compared to patients who lack STAG2 expression. So if you think back to that simplified clinical problem I presented to you earlier, what we're hoping one day is that perhaps STAG2 evaluation can be that biomarker that will help clinicians to guide their clinical management and their treatment protocols for the patients. Of course, there's a lot of work to be done, but we're really, really excited with the potential that we think this biomarker has. And so where do we go from here? So one thing that we're looking at is perhaps an antibody development, generating a peptide antibody. The antibody we use is obviously proprietary, so we don't know what region of the C-terminal it targets. And one important point that was actually brought up by one of my committee members was to determine if there's actually any S1A or STAG1 cross-reactivity. Unfortunately, in all the papers I've searched, there's 3 papers that look at STAG1 and STAG2 in conjunction, and they do not use my antibody. So that is something I will be pursuing. So I actually played around a little bit with trying to align these proteins, very new at it. Not sure if I did it completely right, but what I did find is that there's about 70% homology between STAG1 and STAG2. So this is definitely something that I am eager to kind of start to tease apart. We are also now looking at the role of STAG2 expression in terms of prognosis in muscle-invasive bladder cancer. So again, we've partnered up with Lars and Aarhus University, and there is some data looking at STAG2 status and how it relates to outcomes in muscle-invasive bladder cancer. But there's only 3 studies we know of, and there's discrepancy in the results. So we are very excited now to have these TMAs in our hands. They're stained, uploaded, and ready to start sciencing on. And then finally, I've mentioned this in a few of my other talks, the next logical step with something like this would be a prospective study. We have made some very valiant attempts to partner with the BWELL study. They are poised to be the largest bladder cancer study looking at predictive risk factors in the country. There's a $1 million grant and it closes May 2019. Ran into some issues because they did not have the foresight to collect tumors along with their studies. But we think it might provide a robust resource for us to not only look at STAG2 in terms of a cohort that's very well clinically annotated, but we will also be able to have the chance to study STAG2 in a longitudinal fashion and see what happens to STAG2 status, if anything, over time. And again, just some more pretty slides. I really love these guys. Okay. So now let's talk about trying to figure out what's actually going on. What is STAG2 actually doing in bladder cancer? So the second aim of my thesis proposal was to characterize the phenotypic differences in STAG2 wild type versus STAG2 null or STAG2 mutant cell lines. And I hypothesized that STAG2 tumors were less aggressive and more susceptible to therapeutic strategies due to aberrant mitosis. So in order to do this, I first set out to develop isogenic pairs of cell lines using knockdown methodology. So these cells would differ only in STAG2 status. And then with the ultimate goal of utilizing the model I developed to start to characterize some of these phenotypic differences. All right, so what do we know about STAG2? We do know that mutational activation may have an adverse effect on proliferation. So here we see ACT116s, which are colorectal line. And in this case, they actually knocked in, this is some of Todd and JS's previous work, JS actually knocked in tumor-derived mutations and found that proliferation had slowed. And in 42 MGBA and H4 cells, which are both the GBM lines, they corrected the mutant allele and found that the corrected lines had increased proliferation. And then we've also seen in some other literature that in upper tract urothelial carcinoma, loss of STAG2 is significantly correlated with lower Ki expression. Or I'm sorry, Ki67 expression. We also know that tumor-derived nonsense STAG2 mutations result in a reduction of sister chromatid cohesion integrity. So in this experiment, JS6 cells in mitosis with nicotazole, and they looked at the pro-metaphase spreads to analyze sister chromatid cohesion. So on the top panel, you see what normal sister chromatid cohesion looks like. And then in the right panel, you see parallel chromosomes, which is what you see when cohesion is lost. And then they found that tumor-derived nonsense mutations resulted in a decreased integrity of sister chromatid cohesion, while interestingly, tumor-derived missense mutations resulted in wild-type sister chromatid cohesion. STAG2 truncating mutations also result in increased chromosomal lagging and reduced anaphase integrity compared to wild-type STAG2 cells. So firstly, proliferating cells were fixed and stained, DAPI stain, ACA for ACA for centromeres. And then they determined the fractions of anaphase cells with chromosomal missegregation errors. And in this particular case, there were actually 3 cell lines with knocked-in tumor-derived mutations that significantly increased chromosomal lagging. And lastly, we've also seen that glial GBM cell lines containing STAG2 mutations are more sensitive to PARP inhibition. So again, the H4 and 42 GMBA lines are STAG2 mutants, and the knock-ins are the corrected allele. So what you actually see here, where the arrow is pointing to, that is the non-corrected allele, and correcting the allele, as you can see, decreases sensitivity. And then these are just some other PARP inhibitors that they tested to demonstrate that the trend did hold. All right, so setting off to develop my model. So in order to start off with this STAG2 deficient model, I first employed CRISPR in order to get the knockout, thinking it would make a much more robust model system with which to study. And so just to back up a bit, previously our postdoc, Victoria Hill, had generated a corrected STAG2 knock-in line from a cell line called VM-Cub3s using AAV-mediated somatic gene cell targeting. So on the bottom, highlighted in red, is just the cell line that she had worked with. And so initially she had to design homology arms for the coding exon 4, which is where this mutation in VM-Cub3 lies. And then briefly, she infected the cells with the virus, selected with drug, performed a limiting dilution. Diagnostic PCR revealed that there were 6 knock-in positive colonies, and then sequencing validated 3 of these. Finally, pre-infection removed the flox glycite acceptor, second round of limiting dilution, and then a Western blot to ensure that expression is contact. And here, number 317 is our corrected VM-Cub3. So then I went to go test STAG2 expression on a panel of bladder cancer cell lines to start collecting lines that could be used to develop this model. As you can see, we have HeLAs, which is just our standard to compare to, obviously not a bladder cell line, VM-Cub3s, the knock-in, and then these 6 other bladder cancer cell lines. And here's a little just description of the little we know from ATCC regarding these lines. All right, so my workflow for CRISPR. First, infect, select, and expand my cells. Then with the pools, confirm loss of STAG2 expression with Western blot. And simultaneously, once these cells are expanding, I performed a limiting dilution assay in order to grow up clones. Once the clones were growing up, I would expand them, perform a Western blot to confirm loss of STAG2 in the clones, sequence to confirm the knockout, and yay, science. In theory. All right. So I started off with those 6 cell lines we spoke about. J82s and HT1397s were very, very difficult to work with and were dropped. T24s and TCC stuffs actually did not survive expansion. And even after repeating it, for some reason, they were just not very happy campers. But 5637s and SW780s pulled through, and I was able to confirm loss of STAG2 expression, or at least reduce STAG2 expression in the pools. Okay, so while my pools were growing up, I got a little antsy and I wanted to start just doing some experiments to see if anything was going around. And so, as I alluded to, we've seen that GBM cells have increased sensitivity to PARP inhibitors. There's also been some literature that suggests that these cells might just be more sensitive to chemotherapeutic agents owing to increased mutation burden and the DNA damage effects, et cetera, et cetera. But in my VM-Cub3s, I didn't see much of a difference. And then when I went to go look at the 5637 pools, again, no difference. And I was like, 30-year slump was starting to hit. I was, what's going on? And again, in the SW780s, the same story. But what was really suspicious to me is how beautifully these laid over each other. So I wasn't really sure or confident that the knockdown was hanging around like I wanted it to. So what we actually saw is that over time, STAG2 expression is actually not maintained as passage length increases in STAG2 knockout pools. So we thought really hard about this and tried to brainstorm why, and a few things came up. So first of all, there's the fact that CRISPR didn't work well in every cell. Yes, STAG2 is on the X chromosome. There's a chance that, you know, these cells are not euploid, so we might be missing some alleles. Is there actually a proliferative advantage? Is that actually what's going on? And that was what I really thought was happening. But then with the knowledge of some experiments that JS had performed, we got a little bit smarter. So essentially in HeLa cells, JS was able to create and grow up STAG2 knockout clones. And they grew for a while until they didn't. And they just essentially petered out. So what we suspect is that STAG2 loss is lethal, but maybe not right away. So here's the summary of my STAG2 knockout attempt. We were able to obtain pools. Unfortunately, no colonies grew up. So this was actually a frowny face for most of that time. All right, so if the knockout didn't work, let's try knocking down. So we still have some levels of STAG2 protein hanging out in the cell. My shRNAs work beautifully, as you can see here, especially shRNA2. But again, similar to the CRISPR story, while I was able to confirm the loss in pools like we just saw, I was not successful in growing up the colonies or in growing up the clones. So then I started to think, well, maybe these cells are just not amenable to cloning. So to get around that issue, I decided to move into a transient system. So here's just a preliminary screen of a handful of saRNAs against STAG2. And as you can see, they work beautifully, especially at 72 hours. And then I went and tested them in the lines I really cared about, my bladder cancer cell lines.
Sorry, can I interrupt you? Because this has been on my mind for, so because basically you have shown that this there is DNA damage or likely DNA damage that is being induced, right, when STAG2 knock down. So the main question to my mind is they don't grow because they're activating some checkpoint that, you know, compensate for that DNA damage.
Yeah.
P53 being, you know, probably the most likely culprit. So have you tried to conduct a knockdown, a knockout, selecting cells based on downstream checkpoint inhibitors such as P53?
We haven't done that, but we have.
I bet you that they are not surviving because you knock him down, P53 gets activated, so eventually they stop growing or they die.
So we did actually see in the 2013 paper, David saw that there was a trend where tumors that lacked STAG2 actually had concurrent P53 mutations or overexpression.
Based on everything that you have shown.
And also, there was just a paper that came out with, they're looking at Ewing sarcoma mutation burden with that, and they've already shown that P53 and STAG2 mutations correlate or actually predict for lower overall survival and time to progression.
In my opinion, you want to have a model to study proliferation or cancer, progression, even you have to get rid of it somehow.
Yeah.
That it says to grow somewhat. That's my.
No, that makes sense. Okay, so these are just the effects of the saRNAs against STAG2 in the bladder cancer cell lines. Again, BMCUB3 lack STAG2 expression. That's why I don't see anything here. And then we added H-black cells and H-tertiary cells to this panel. So these are actually immortalized primary cell lines that we were very happy to come across into the literature, and started working with these so that we would develop a control as well. All right, so I think where I left you last time was somewhere about here, that we had started trying to develop a model system, finally working to got into the transient system. And now I can hopefully start playing with some of the real experiments that I wanted to do. So I know that STAG2 has an important role in mitosis. JS was becoming an expert at the confocal and making cells green. And my transient model system was working. Like any grad student, good grad student, I know where free food is, and I know how to use my resources. So I decided to image cells to determine if there's a mitotic phenotype due to STAG2 suppression. So this is an example of a really great model system for looking at cells under the scope. These are HeLa cells that are tagged with GFP on the H2B protein. And just to give you an idea, they move pretty slowly, divide pretty nicely, regularly, so you can see them really well. And this is a clonal line, so also just note how uniform the brightness is, which, as you can imagine, is very helpful in terms of analysis. But of course, these are HeLa cells, and I am determined to get this to work on my model system. So here's my workflow. Infect the cells of the GFP virus, collect the pooled clones, also perform a limiting dilution to try and grow up these clones. And then we get to the experiments with the siRNAs and the live cell microscopy. So unfortunately, with the first virus I tried, we found that the cells were way too faint, and they were just not amenable to this type of experiment. But thankfully, with this second virus, I was able to generate pool clones that were very, very nicely bright. And I got a little bit smarter this time, gave up on limiting dilution, and finally went to clonal methods. Sometimes the old school ways are just the way to go. And I am thrilled to say we were able to generate many, many clones. So now we have GFP positive clones for the VM CUB3 isogenic pair, T24s, and also for the 2 primary lines. I was really happy. I had 41 cell lines in culture, and I literally took a picture and sent it to my mom. All right. So this is where I started to use some of my cells and start to play with the live cell. So this is a clone of the VM CUB3 line. And what I want you to appreciate is that these cells have much fewer mitoses than the HeLa cells. And each mitosis takes roughly about 2 hours. And you see about maybe 16 cells of HeL. And this is actually a 10-hour clip. So over all 10 hours, there are only about 5 mitoses. Whereas in these H black lines, and this is a pool, so you'll see how different the intensity and the brightness is. These move a ton, and they have much fewer mitoses, although they happen much faster. They take under an hour to divide. So at this point, my goals are I still need to work on optimizing this, the live cell protocol, and just getting that system to work. And another issue I ran across is that we actually find that these cells are particularly sensitive to the transfection reagent. So I transfect immediately before placing the slide onto the scope stage. And what you see is in the non-transfected cells, they're happy as can be, but the negative controls are already looking pretty unhappy, to the same effect that the cells, the wells containing the siRNA are. So we need to tease that apart a little bit and test a few different reagents. And there's also just some new literature that has come out. I mentioned the Ewing sarcoma mutational burden signature. There's also a paper that came out, Todd's former student was actually on it, that showed that STAG2 loss actually led to resistance to viral infection by instigating the JAK-STAT pathway and then finally interferon stimulated gene expression. We've also come across literature that has suggested STAG2 silencing in normal human cells that lack telomerase leads to increased recombination of telomeres, delayed telomere shortening, and postpones senescence, leading to the hypothesis that perhaps STAG2 loss in normal cells gives these cells more time to develop mutations, again, leading back to that mutational burden hypothesis. And then finally, there was a new paper that came out just a few weeks ago that showed repression of transcription at DNA breaks actually requires cohesin through interphase. And this is distinct from its role in regulating sister chromatid cohesin. And they've actually ID'd several cancer-derived mutations that support sister chromatid cohesion, but cannot repress transcription. So initially, a lot of the thinking with perhaps the way SAB2 was involved in cancer was that it was leading to aneuploidy because of the unfaithful sister chromatid segregation. But we don't believe that's the case anymore. And we think it really lies in either genome organization or something, some kind of transcriptional repression. All right, so before I get to my concluding remarks, just to show you my rigor and reproducibility, happy to go back to that and answer any questions, but just in the interest of time. All right. If you have blood in your urine, go to the doctor. Go to the doctor. It is never okay. I came across this email, one of the very first emails I wrote on this project. So I hope I've convinced you that this program has really helped me to grow as a scientist, but also as a human being. I've really found my voice, other passions for science communication, advocacy, beating up anti-vaxxers, you guys all know that. But I've really gained a lot. Gray hairs, but I all lost the thumb, so it evened out. Okay, sharing is caring and communication is key. All right, so this is what our project has actually led to, and it all started with Dr. George Phillips approaching Todd because he saw that initial Kaplan-Meier curve of the preliminary cohort that David had studied, and saw that he was, STAC2 seemed really interesting, and lo and behold, they started a collaboration. But Georgetown, besides Dr. Lafredo, who deals with bladder cancer in a little bit of a different context, we didn't really have many people here who studied bladder cancer exclusively. We didn't really have the resources here. So of course, that's the project I took on. But I'm very, very proud to say that over the course of the last few years, we've established a multi-institute network, kind of like what Todd had alluded to, that spans the globe, is still growing, and hopefully, we're starting to get our foothold into this bladder cancer world. Like I said, it's grossly understudied in the U.S. We've got a lot of work we need to do, but I'm very, very hopeful that this is a very big start for Georgetown at large. Lastly, almost not lastly, but there's also knowledge to be gained in failure. As you noticed, my whole aim too was a whole bunch of this didn't work, that didn't work, this didn't work, but we learned so much from it. For example, with the HeLa cells petering out, there's still something to be gained from there. So that was a long, tough lesson to learn, but that's science, and that's why we're here, right? And lastly, I never had to autoclave until I was 29 years old. So you don't have to autoclave. It scares me, and there's ways to get around it. All right, and then lastly, to thank everybody, well, it's my name on the dissertation. This work is hardly the result of just my singular efforts, and it would not have been possible with the people listed here, and besides, obviously not even a complete list. Well, firstly, I'd like to thank Todd for accepting me to the program, and then accepting me to this lab, and then giving me a project that no one had done and trusting me enough to kind of run off with it. Oh, and also for banning the word data meeting in our lab, because apparently if you cry in front of your boss, he won't say that phrase anymore. Of course, I'd like to thank my committee members for their time and thoughtful critiques. Our collaborators with whom this project would have been possible, the whole department, Jaden for reminding me about all the signatures I had missing all the time, my funding, the tumor registry, TL1, and then last but not least, the histology core. I affectionately call them my histology dream team. Without them, this project would not have worked, but besides the scientific contributions they've made, they have really become great friends and great supporters, so thank you. And unfortunately, my family couldn't be here today, but they also are a very big part as to why I'm here. So with that, I will open it up to questions.
Oh, nice talk. Thank you. So I learned from your last talk from you and Todd that other molecules in the cohesion complex can be mutated in a non-overlapping way in STAT2. So you were able to use those in addition to STAT2 as a biomarker that might increase the sensitivity of them as a biomarker. And
so that's one thing we're definitely looking at, especially because the group in Denmark, they've done a lot of work with various different molecular markers. The other thing is, actually, I think I have the slides here. So we actually do have sequencing data on one of the cohorts from Denmark. In one of the muscle invasive cohorts, we also have a subset of patients who have whole exome sequencing, I believe. So this trend based on the sequencing data wasn't statistically significant, but what's important to note is that this isn't stratified for mutation type, and we know that nonsense mutations and missense mutations behave really differently. So actually, in fact, with the IHC assay we use, we suspect we're also picking up missense mutations. So I would imagine with sequencing, we might be able to separate that curve a little bit better.
Very nice data, very difficult to present.
Thank you.
Congratulations.
Thank you.
For this study here, I don't know, you may have said it, maybe I missed it. So when you have this separation between the STAT2 high, STAT2 low, those that report and that don't report, is that an evolution from one stage to another, meaning you have tumors that initially have STAT2 expression and then suppress it, or do they start as two completely molecularly different?
So yeah, that's a great question. We have actually looked back into the Georgetown cohort to see if we have any like secondary tumor samples. We don't have tumor samples on site. We had one patient where I think we had lung aspirate from his metastasis, but that's kind of why I'm hopeful with the BWAL study, that prospective study, because they will have been following these patients for so long. I think it's super important that we get that longitudinal story to see if there's any evolution.
What's the precursor lesion for bladder cancer? Is there a precursor lesion, like a early stage?
So there's between the papillary and the carcinoma in situ tract development, there's characteristic mutations in there. In the papillary, FGFR3 is sometimes cited as the instigator.
if I were a patient, I'm not sure I will be completely satisfied with having transurethral resection only, because you can't really evaluate whether the smooth muscle is invaded or if the invasion goes beyond that. What happens when?
What do you mean you can't?
Because you go in, you just resect the tumor from the lumen.
They go all the way into, you get muscle, they resect all the way deep into the muscle.
Yeah. Okay, and then if you suspect it goes further, then what happens? They actually do a second surgery or what?
Yeah, so they will do what's called a secondary restaging turbid. So yeah, if they're not satisfied that, say, they don't see muscle on that, then they won't comfortably call that as non-muscle invasive. They will do a secondary turbid.
I'm just interested in what you think the driver mutations are in bladder cancer and how they might interact with this STAT2 mutation expression loss. Because is there any idea of you guys played around with the database to look at whether the people who relapse with the STAT2 mutation may have a different driver mutation?
Oh, that's an interesting. No, we haven't looked at that, but that's
because your argument, I think, that STAT2 wild type is working with something else with bad news.
Yeah.
Because in its own it can't be a driver, right?
Exactly.
It's wild type. So I just wondered if you guys sort of had a drink.
No, to be honest, not to really start really delving deeply into that. I mean, I'm intrigued a little bit by that paper that, had cited that, perhaps the STAG2 loss early on just gives the cells more time to collect mutations. I know that not in non-muscle invasive bladder cancer, but in muscle invasive bladder cancer, it has one of the highest mutation burdens up there with what is it? Lung and skin, I think, are the two high ones. But in any case, so yeah, I don't have a great answer for that. Good. Yeah.
Oh my God, everybody be quiet. You just keep showing that. Penis smile.